Is my screen visible?
Yes, sir.
What is on screen?
OneNote.
OneNote. Okay. Okay. So what we discussed in the last class? I think we solved some problem, right? PHPK. Somebody can respond.
Sir, we saw through the synthetic drug processes and stuff.
Oh, so okay. I completed the sources of leads, right?
Yes, sir.
Okay. Okay. So as of now, we'll move to the second unit. So second unit is completely meant for pharmacokinetics.
Sir, sorry to interrupt you, sir. Can you just revise what we saw in the last class? It's been a
Okay.
1 week
Okay.
break. Thank you, sir.
Sorry.
Can you revise through the last class, sir?
Okay. You need revision for last class. Okay. Okay. Before that, can you check your OneNote? So I try to sync my OneNote with your class notebook. So can you check your OneNote? Let me know whether the content is updated. So go to the notebook, go to content library. I just want to ensure whether the sync is working correctly. Go to the medicinal chemistry notebook. You have a section called as content library. Go to that.
Yes, sir. Your current screen is
Yes,
CCA,
right.
sir.
Sync, right? So okay, that's fine. Okay, so so hereafter whatever I am writing here, that will be automatically synced to your notebook also, right? You can use it as a reference material. Okay, the revision of previous lecture is like we discussed about the various sources of hit molecules. Various sources of hits, leads, and candidate drug. So I hope that you know the difference between hit molecule, lead molecule, and candidate drug. So we have three important sources. The first source is the natural source, and the second source is the synthetic, and the third source is de novo. So this de novo ligand design is also called as computer-based drug design, or computer-based ligand design, or we can say it as structure-based ligand design. All this refers to the same. Okay, so it is de novo ligand design. So these are the three major sources of hits, leads, and candidate drugs, which means that so these are the sources from which we can get hits, leads, and candidate drug. So initially we have to look for the hit molecules. So we should have enough number of hit molecules, because during the pipeline of drug discovery, in every stage, every step, some amount of the molecules will be failed. Okay, so we have to start with large number of hidden molecules to ensure that we have enough number of candidate drugs which can be further taken into the clinical studies. So these are the three major sources of hidden molecules. I gave a couple of examples for how we can use natural compounds as a drug molecule. There are many drug molecules which are approved by FDA, which are obtained from natural source. So natural source means it can be either from plant, animal kingdom, or marine. Many things we can say, right? So all those comes under natural. So very good example is morphine. So morphine is a very well-known drug molecule which is used as a painkiller, analgesic molecule. So this morphine is directly obtained from a plant called as a poppy capsule. If you remember, I have shown the the plant itself, right? So the source is poppy capsule. So directly we can isolate and use this morphine as a drug molecule, which was approved by FDA. So sometimes we can make use of natural compounds as starting material for designing drug molecule. So I gave a couple of examples. One simple example is using salicylic acid for synthesizing aspirin. So aspirin is a FDA approved drug molecule. We cannot obtain aspirin as it is from any natural source, but the starting material for synthesis of aspirin, it's a one step reaction, which is salicylic acid, can be obtained from plant source. Okay, so those are examples for how we can make use of nature as the source of hit molecules or lead molecules or candidate drug. So coming to the synthetic, sometimes you may not be successful, going with the natural nature as the source. So sometimes you may need, the molecules which are not naturally available. So in that case, we can go with synthetic. So there are two broad ways we can make use of synthetic chemistry for identification of hit leads and candidate drug. So the first approach is very normal traditional organic synthesis. So in which what we normally do is we take reagent A and we let reagent A to interact or mix react with B. So that will give the product C. So this is what I mean as normal traditional organic synthesis. So basically, so then the product of the first step C can be allowed to interact with a D in the second step to give E. So the product of the second step is E. So it can be multiple steps. Each step will get only one compound as the product. In the first step, the product is C, and in the second step or second stage, the product is E. This is what I mean here as normal traditional organic synthesis. Very well, we can use this for drug discovery purpose, right? So as I said, salicylic acid, okay, so acid dehydration will lead for aspirin. It's a one-step process, right? So this reaction is an example for normal traditional organic synthesis. So there's another method very well used for drug discovery process is called as combinatorial library design, combinatorial or parallel synthesis. We can say this is a kind of advanced synthetic chemistry method uh, carried out in chemistry labs. So the main difference between the normal traditional and this combinatorial or parallel synthesis is like, so in case of combinatorial and parallel synthesis, okay, the final reaction vessel, say for example A plus B gives, say this is the final reaction vessel. So the final reaction vessel won't be having only one type of final product. It may have many types of final products, like say uh, GHI. So here GHI or individual molecules. So it can have mixture of different types of molecules. So what we do here is A plus B, say A plus B plus C plus D. Okay, so all the reactions are taking place in one single vessel. Hence the final reaction vessel, it may have more than one type of molecules, and that is what is called as a combinatorial or parallel synthesis. So we have to discuss about this in detail. We have separate unit for this. We have separate topic for this in fourth unit. So while discussing the fourth unit, we will see what are the various approaches which are commonly used in combinatorial and parallel synthesis. We'll also discuss about what are the advantages and disadvantages of going for combinatorial and parallel synthesis, right? So as of now, so combinatorial and parallel synthesis are sources of drug-like molecules. It may be hit molecule, lead molecules, or candidate drug. So synthetic chemistry is the second one, promising way to get the hit lead candidate molecule. The third method is called as the de novo ligand design. So the basic idea of de novo drug designing is like it's a kind of in silico approach. in which we should know the 3D structure of the drug target for which the drug molecule should be designed, and we use some computer algorithms to develop the drug molecule based on the architecture of the binding site. So if you do so, that is what is called as de novo ligand design. So in summary, the previous lecture is, what I discussed was like kind of what are the various ways by which we can get the hit molecules, lead molecules, and candidate drugs, or we can say the various sources of hit molecules, lead molecules, and candidate drug. So this is what exactly I have discussed in my previous lecture. Any question up to this I can address because long gap, any question up to this I can address before going for the next topic. Hope everybody is connected, right? Am I audible?
Yes, sir.
Yeah, now it's
Yes, sir.
open for question. We can have some question and answer session, right? Because it's a long gap. Any question I can address. It can be from any topic, doesn't matter, right? So some of you might prepare during this holidays. No, any doubt related to this subject, I can answer that before going for the next topic. Any questions? Okay, I assume that no questions. Can someone check the class notebook now whether it is syncing? Because during the holidays, I just try to sync the notebook.
Yes, sir. It's synced.
It's syncing, right? So okay, perfect. Okay, so no issues, right? So a few topics I left in first unit. I'll come back to that. I won't skip any topic, right? So as of now, I just wanted to move to the second unit. So the second unit is pharmacokinetics. Already we discussed the basics of pharmacokinetics and pharmacodynamics and drug action, right? So let me again repeat that. So three important definitions. One is called as pharmacokinetics. Second term is called as pharmacodynamics. And the third one is called as the drug action. Maybe kind of repetition, but we cannot skip this. So first, let me tell what is meant by drug action. Okay. So drug action is, basically, the difference between the drug molecule and drug target is the drug molecules will be always smaller in size. So we discussed about Lipinski's rule of 5. He stated that for a good oral molecule, drug-like molecule, the molecular weight should be in the range of 500 Dalton. Okay. So based on this value, we can say that always the drug molecules will be, most of the time, not always, most of the time, the drug molecules will be having a smaller molecular weight comparing to drug target. So what are drug targets? So drug targets are the macromolecules, which are basically present somewhere within the body or within the cell. In some way, they are responsible for causing the disease or some way related to the disease, right? So those are called as drug targets. So there are 4 classes of molecules that they can act as drug target. Proteins take the first position. Why I say this is because almost most of the drug molecules which are approved by FDA, they target any of the protein molecules which are present within the cell or within the body. So proteins class of molecule can act as drug target. And next is DNA or RNA, or we can generally call it as nucleic acid. They can act as drug target. And we can say lipids, sometimes they can act as drug target. Or we can say sometimes glycoproteins or carbohydrates. Okay, so these are special type of proteins in which the carbohydrate is normally embedded within the protein molecule or lipid, whatever it is, right? So carbohydrates, they can act as a drug target. So the specialty of all the 4, right? So the uniqueness of all this 4 or all these 4 classes of biomolecules, they are called as macromolecules. We cannot say it as a small molecule like drug. So if we compare the molecular weight now of the drug molecule to that of drug target, obviously the drug targets will be having very larger molecular weight comparing to the drug molecule. Okay. Second point is that, So why, these are not the only macromolecules which are present within the body or cell. We can say, even more number of macromolecules within the body, but these are the molecules which are very suitable to act as a drug target. The reason for this is because protein, DNA, RNA, lipids, carbohydrates, they're druggable. In one of my lecture, if you remember, I talked specifically about druggable. Okay, so which means that these are the class of molecules. They naturally they have some space for binding the incoming drug-like molecule, right? So, say for example, the drug molecule should go and bind to a receptor. It can be a protein molecule or DNA, RNA, or lipids or carbohydrate, whatever, right? So I generalize this as a receptor. Okay, I generalize this as a receptor. Okay, a drug molecule should go and bind to drug receptor, where it should form a drug receptor complex, and the formation of this drug receptor complex results in the biological response. And this response and this entire thing is what is called as the drug action. So drug action is how the drug molecules binds to the drug target, and how it forms the drug receptor complex, how that leads for the biological response. Specifically, the final biological response is the drug action. Okay. So coming to pharmacokinetics and pharmacodynamics, so these are the two major, wings, or two major studies of pharmacology. Okay, so mainly we study pharmacokinetics and pharmacodynamics of drug molecules and pharmacology. This study may be either in a animal model, or it can be in humans. So it can be either in a preclinical level, or it can be in a clinical level, right? So a very simplest definition for pharmacodynamics is, it's a very layman definition, not the exact scientific definition. So pharmacodynamics is what the drug does for the body, right? So let me write it here. Okay, not the textbook definition, just for the understanding what the drug does for the body. Already we defined many times, and this is what is called as pharmacodynamics, right? So what is a pharmacokinetics? What just the opposite of this, what the body does for the drug molecule is what is called as pharmacokinetics, right? So second unit, as I said, is completely meant for pharmacokinetics. So what you're going to learn is kind of, what are the various. So we start with very fundamental definitions of absorption, distribution, metabolism, excretion, because these are the four important properties which are studied under pharmacokinetics. And we'll discuss about how the various route of administration affect absorption, distribution, metabolism, excretion. We'll see that in very detailed manner. And what are the various factors which affects absorption, distribution, metabolism, excretion, that you'll be studying. And what's the need for this pharmacokinetic study, how this study is going to help for the drug discovery. So that is the entire story of the second unit. Okay, that's what exactly we'll be studying in the unit 2, right? Let me share a presentation. Is my presentation visible?
Yes, sir.
Yes, sir.
Yeah, okay, yeah. So this is what exactly, I just wanted to cover in the first half of the second unit. Okay, so I just wanted to introduce what is meant by pharmacokinetics. So general classification of pharmacokinetic properties like ADME, absorption, distribution, metabolism, excretion. So we'll see what is meant by drug regimen, what exactly is the definition for drug regimen, and importance of pharmacokinetics in drug discovery. This is very important, right? So why actually we have to study this pharmacokinetic? What's the need for studying about absorption, distribution, metabolism, excretion? We'll learn about that. So drug concentration analysis, its therapeutic significance is kind of numerical things. We are going to solve some problems related to that. And then we will see about pharmacokinetic models. We have different types of pharmacokinetic models. Whenever we do any kind of pharmacokinetic studies, we have to fit that to some model to ensure what type of kinetics is followed by that drug molecule. So we'll see what are the different types of kinetic models which are available. And So mainly I'll be talking about how these factors will be affected if we take the drug molecule as a oral drug molecule, if the administration is oral administration, or if we change that to intravascular administration, how the parameters will get changed, right? So these are the primary thing that we'll study in this unit. So then intravascular administration. Okay, so then intravenous injection, how the parameters will be affected if we go for IV injection. So then we'll see about elimination and elimination half-life. These are very important parameters to decide what dose should be administered to either the animal model or to the human system. Clearance and significance, because they also play important role. They also decide the dose. For fixing the dose, we should be very clear about the clearance and how that is happening. So if we give the drug molecule as intravenous infusion, what happened to the pharmacokinetic parameters, extravascular administration, right? So these are the primary things that we'll see in this topic, right? Okay, so this is a very simple slide. I have a beautiful image on this. So any drug molecule should be administered with a recommended dose, right? So we cannot go beyond that dose. For any drug molecule, there should be some dose. Okay, that is the allowed dose for using it either in the animal model or in the human system, right? So this dose will differ from using a smaller animal models and larger human system. And if I say that human system, okay, one point I just want to make, that the average weight of human male is considered as 60 kg, right? So whatever values here I mentioned, that is approximated for 60 kg male. Okay, so we take some dose of the drug molecule. Say you have some pain and you are taking aspirin. Aspirin is very well known drug molecule for reducing the pain. So once if you take some dose of the drug molecule, so immediately after taking the drug molecule in any of the administration, it can be any method, right? So the drug molecule can be taken as a oral drug molecule, or we can go for intravenous injection or intramuscular injection, right? So in any way, if you take the drug molecule, so what happens is the drug molecule first enter into the body. We already discussed about this while talking about the journey of the drug molecule, if you remember, right? So the entire body is not having the pain. Say for example, this person is having some pain on his hand, okay, so, near the finger, right? So what happens is the the molecule is taken throughout the body through the process called as distribution, and some quantity of the drug molecule will definitely reach the place where the pain is there, and that site is called as a site of action. That is the place where the drug molecule should go and go and act on the corresponding drug target. So once if you take the drug molecule, once if the molecule is distributed throughout the body, once if some amount of the drug molecule reaches the site of action, okay, so it can be resulted in two different things. Now one is the therapeutic effect. So here, what is the therapeutic effect in this example? So in this example, the therapeutic effect is
Reduction of pain.
it should reduce the pain. Okay, absolutely right. So the person should feel that the pain is somewhat reduced because of in taking the drug molecule. That is what is called as the therapeutic effect of this aspirin. So but none of the drug molecule will only produce the expected therapeutic effect. There are more chances for the drug molecule to interact with some other part of the body, or in other words, some other targets. So those are called as off target. Okay, so the meaning of off target is that the drug molecule is designed for certain target, but it is not necessarily the molecule should go and bind only to that specific target. There may be lack of specificity for the drug molecules. So because of that, the molecule can go and interact with some other unaffected cells which are not injured, or it can go and interact with some other drug targets, or some other macromolecules which are present within the cell. Those are what are called as off target effect. So if any molecule is having off-target effect, then that may lead for another type of effect called as toxic effect. Okay, this is a very general thing that may happen once if you take any drug molecule, irrespective of aspirin. So whenever you take the drug molecule, immediately the pharmacokinetics and pharmacodynamics comes into picture, right? So the effect of pharmacodynamics is the therapeutic effect and the toxic effect. The effect of pharmacokinetics is how the drug molecule reaches the site of action. That is what is called as the pharmacokinetic effect. It's a very fundamental thing, right? So we will explore more in the next slide. So what is meant by therapeutic? So I gave few definitions for the therapeutic. So therapeutics is in order to produce the therapeutic effect, drug must have sufficient concentration in the targeted tissue. This point is very important. Again, I go back to the previous slide. You are taking some drug molecule, and if the concentration of the drug molecule at the site of action is not sufficient, which means that we cannot expect the expected therapeutic effect. So the concentration at the site of action is very important. So how much amount of the drug molecule is present near to the site of action? Okay, the site of action is the place where the drug molecule should act on to produce the response or drug action. So how much amount of the drug molecule which is present around there, that plays a very important role. So that is what is given. So to produce the therapeutic effect, drug must have sufficient concentration in the targeted tissue. Okay, it is not about how much amount of the drug molecule is taken. It is about how much quantity of the drug molecule or how much concentration of the drug molecule is present near the targeted site. That plays important role. That is going to decide how much amount of the drug action we can expect out of that drug molecule. So what I mean here is again and again I'm stating the same point in different way, that the concentration of the drug molecule near the site of action should be as large as possible. Okay. So there's a direct relationship between the therapeutic effect and the concentration of the drug molecule which is present near the site of action. Okay, the relationship is very straightforward. If the concentration of the drug molecule near the site of action is larger, then we can expect larger therapeutic effect. So it's having the direct relationship, right? So this is a different way to define what is meant by pharmacodynamics and pharmacokinetics. Pharmacodynamics, it deals with concentration effect component, and pharmacokinetics that deals with dose concentration. Okay, no need to worry about anything, right? So even if you don't understand, no worries, right? So I have many number of examples to make it understand. As of now, therapeutic is something related to how much concentration of the drug molecule is present near the site of action and how much the effect is produced by the drug molecule. That is what is called as therapeutics, right? So there are four important parameters which are commonly studied again and again. I am repeating this point under pharmacokinetics. That's called as absorption, distribution, metabolism, and excretion. Okay, these are the four parameters which are normally studied under pharmacokinetics. Okay. So here again, I gave the definition for pharmacokinetics. All right. So pharmacokinetics can be defined as what the body does for the drug molecule. Okay. So these four important parameters can be classified into two types. One is called as translocation process and another one is called as transformation process. Okay. So this is a physics term, translocation and transformation. The term itself says that translocation is movement. So movement of the drug molecule from one part of the body to other part of the body, that is called as translocation. And transformation is conversion of the drug molecule in its original form into some other form. So we can say that absorption, distribution, metabolism, excretion, all this could be either a translocation process or transformation process, any one of that, right? So among this, absorption, distribution, and excretion, these are example for translocation process, which are related to movement of the drug molecule from one part of the body to another part. And transformation, metabolism is an example for transformation, because in metabolism, the drug molecule in its original state will be converted into some other state or some other form, right? Okay, so I skipped few slides, not much important, right? So coming to pharmacodynamics, as I said, pharmacodynamics is what the drug does for the body. Okay, so once if you take the drug molecule, you are taking the drug molecule for cure, for treating some diseases, or if you have some pain or whatever it is, illness, right? So we take some drug molecule. So once if you are relieved from that problem, it can be a disease or pain, whatever it is, then that is what is called as a pharmacodynamics, what the drug does to the body, right? So dose response relationship is very important. Again, I repeat that we cannot take, there are kind of restrictions in taking the concentration of the drug molecule. So once we take the concentration beyond that recommended concentration, even the molecule which are very beneficial for the human, it may produce a lot of toxic effects, right? So that is what is called as dose response relationship. So for any person that we have to fix the dosing regimen, okay, this dosing regimen will differ from one person to another person. Okay, so it's not going to be same. For most of the people it may be same, right? But it may slightly differ from one person to another person. The physicians will decide the dosing regimen just based on the pharmacodynamics and pharmacokinetic properties. That is why these two properties are very important in drug design. Okay, so if you want to fix the dose regimen, definitely you should have a very good idea about the pharmacodynamics of the drug molecule as well as pharmacokinetics of the drug molecule. So the reason for this is that we always wanted to achieve, maximum response with minimal adverse effect. Again, this slide is very interesting slide because all the drug molecules will produce some amount of adverse effect. We cannot say that we cannot find a drug molecule, or we cannot figure out any drug molecule that has zero adverse effect. You cannot find any molecule. Even if you take aspirin or paracetamol, those molecules will have some amount of toxic effect. Okay, the primary idea of this is that the beneficial effect should be very high comparing to the adverse effect. The adverse effect should be as minimal as possible. Okay, that is what we study under the dose response relationship. So as I said, this pharmacokinetic properties will change if we change the route of administration. Okay, so if I take a drug molecule as an oral drug, its pharmacokinetics will be completely different from the same drug molecule which is taken as intravenous injection. Okay, I'll talk about this in some other slide, right? So the drug route is the journey of the drug molecule. What are the various things that happens? Already we discussed about this in very detail in some other lecture, right? So 4 important things once if you take the drug molecule that happens within the body is, the first step is absorption, and the second step what's taking place inside the body is distribution, then metabolism, then excretion. So these are 4 parameters very often referred to as ADME properties. For a good drug molecule, these properties are very important. They should have good absorption value, they should have good distribution value, they should have very good metabolism property, they should have good excretion property. So if any drug molecule has good absorption, distribution, metabolism, excretion, then we can say those molecules are the candidate drug molecules which can be evaluated in the human system. Okay, so first absorption is, so you're taking any drug molecule, any mode of administration, say oral administration. So if it is oral administration, you're not placing the drug molecule directly onto the blood. The site of administration is mouth. Okay, so the molecule should be taken to the site of action. We cannot directly keep the molecule near the site of action. very rare cases we can do that, not always, right? So once if you keep the drug molecule on mouth, it enter into the it passes the gastrointestinal tract, GIT, enters into the stomach, from where the absorption process mainly takes place. So the molecule somehow getting enter into the blood circulation or general circulation. So this process, the entry of the molecule from the site of administration to the blood circulation, that process is what is called as absorption. So once if the molecule is present in the bloodstream, so you know that the blood is taken to almost all parts of the body, every organ system, right? So every cell, every tissue, entire body is, blood is circulating throughout the body. So wherever the blood goes, the molecules, the drug molecules will be taken along with the blood. So that process is what is called as a distribution. So distribution is the process by which how the drug molecule is taken to the entire body. That is what is called as distribution. So while distribution, the drug molecule, some quantity of the drug molecule will be taken to the site of action, and the molecule will not be taken only to the site of action because the blood is flowing to entire body. So some quantity of the drug molecule will be taken to other body tissues and organs. This entire process is what is called as the distribution process, which means that the molecule will be distributed throughout the body, not only to the site of action. But the therapeutic effect depends on how much concentration of drug molecule which is present near to the site of action. Okay, I'll talk about this later, right? So with some other example. So this process is what is called as a distribution. So once if the molecule is distributed, slowly the metabolism process comes into picture. The molecule, any foreign molecule which we take inside the body should come out of the body through any means. So what happens is the metabolism takes place. The reason for the metabolism is initially the drug molecule will be having many non-polar functional groups. We can say that the balance between the polar and non-polar solvent, the molecules will be highly soluble in non-polar comparing to polar. The reason is then only the drug molecule can penetrate into the cell membrane. So the lipid nature of the drug molecule should be higher comparing to the water solubility nature. But if the molecule has to excrete it in the next step, definitely the molecule should get soluble in water. So the lipid soluble molecule should be converted into slightly water soluble molecule. That is the reason why the nature prefer to metabolize the drug molecule into its corresponding metabolites. So whenever you compare the original drug molecule and its corresponding metabolites, the metabolites will have more water solubility comparing to the original drug molecule. So that process starts. That's also part of pharmacokinetics. So once if the molecule slowly soluble in water, then ultimately the molecule can be easily excreted. It can be excreted through different ways. The main route of excretion is the urine. It can excreted through urine. So whatever molecules which are soluble in water, they can easily get excreted through urine, right? So these are the four important parameters which are studied under pharmacokinetics. So here I have given the definition for each and everything as per the textbook. So what is absorption? Absorption is the process during which a drug enters the body from its site of administration into the systemic circulation. This is what I exactly said. So the movement of the drug molecule from site of administration to the general circulation or systemic circulation, that is what is called as the definition for absorption. So what is meant by distribution? Distribution is the process by which the drug is transferred by the bloodstream throughout the body. So again, if you look at the definition for absorption, so we can clearly say that this process is an example for translocation process. This is an example for translocation process. Because I told that we can classify all the four parameters into two things. One is translocation, another one is translation. So by definition, absorption is definitely a translocation process. It's a movement of the drug molecule within the body. So if you look at the definition for distribution, also we can definitely say that this process is also a translocation process, because as per definition, the process by which the drug is transferred by the body bloodstream through the body, it's a translocation process. So coming to the definition for metabolism, metabolism is a biotransformation. Okay, the name itself says that it's a transformation process, not translocation process. It's not movement from one part of the body to another part. It's a kind of conversion of the drug molecule, change in the structure of the drug molecule in its original form into some other form. So it's an example for transformation process. So the biotransformation process by which the drug is enzymatically transformed into new chemical compounds. So the new chemical compounds which are generated because of the enzymatic transformation, those molecules should be called as metabolite. Those are called as metabolites. So again, the main points related to metabolism is, first thing, it's a transformation process. It's not translocation process. Second is why this metabolism is taking place, because the drug molecules naturally, the balance between water and lipid solubility, if you compare, they will be of more lipid solubility comparing to water solubility. So in order to eliminate the molecule should soluble in water. That's why with the help of enzyme, the nature convert the lipid soluble molecule into water soluble molecule through the process called as metabolism. So whatever compounds which are newly generated with the help of enzymatic transformation, all those molecules should be called as metabolites. Okay. So the last process is excretion. So excretion is a kind of irreversible process, because once if the molecule is excreted out of the body, definitely the molecule cannot be taken inside naturally, right? So unless until you take the another dose. So it is an irreversible process where the drug or its metabolites leave the body. Okay. So there are different ways by which this excretion can take place. We will discuss that in detail in the some other session, right? So these are the four parameters which are very commonly studied under pharmacokinetics: absorption, distribution, metabolism, excretion. Any question up to this I can answer. Am I audible?
Yes, sir.
Yes, sir.
Yeah, any question up to this? Very, very fundamental things, right? So just I explained the definitions. Any question up to this I can answer. Okay, I assume that no questions. If no questions, the next topic is bioavailability. Okay, so we have different parameters along with this pharmacokinetics. We used to study those, right? So one such parameter is called as bioavailability. Okay, the definition for bioavailability is how much quantity of the drug molecule is present within the circulation. Okay, again I repeat, the amount of the drug molecule which is present within the circulation, that is what is called as bioavailability. Say for example, again we take the simple example, aspirin, because it's a very commonly used drug molecule. So the route of administration of aspirin is definitely it's oral drug molecule. So for example, you are taking 50 mg of aspirin as an oral drug molecule. So again, I go back to this slide. So you're going to take 50 mg of this drug molecule. So the dose here is 50 mg of the drug molecule is aspirin. So the question is that if we take 50 mg of aspirin as an oral drug, how much amount of the drug molecule will be present in the circulation? Can you guess? Can we find the entire 50 mg in the circulation, or you have any other answer? Am I audible?
Yes, sir.
I am asking some very simple question that even the sixth standard or seventh standard students they can answer. Should I repeat the question?
Yes, sir.
Okay, so say for example, you are taking 50 mg of aspirin. Aspirin is a well-known FDA approved drug molecule, very commonly available on drug counter. Without prescription, you can get aspirin. So if you take 50 mg of aspirin, so far I explained the different parameters which are studied under pharmacokinetics. So my question is that if you take 50 mg of aspirin, how much amount of this drug molecule I can expect to be present in the circulation if the mode of administration is oral administration? You people have any answer or not? Can someone repeat the question?
Sir, how much will it
Whatever
be in the
whatever question
circulation?
whatever question I have stated, just tell me the question.
Sir, if we consume 50 mg aspirin,
Yes.
if it will be 50 mg in the circulation, that's the question.
So you mean that 50 mg we can find it in circulation?
Yes, sir.
Is it your answer?
Yes, sir.
Okay, I'm not asking your answer. Can you repeat the question first? Hmm?
Sir, if we take 50 mg of aspirin, how much drug molecule will be in circulation, sir? That's the
That is a
question.
question. Yeah, question is right. Can you tell the answer now? Question is right. You're able to see 32 students. Okay. Okay. If you have no answer, there's no point of discussing more.
Sir, 50 mg will be in
No, I'm not
circulation.
expecting an answer from one student. Okay. It's not good sign. I don't know what happened for other 31 students. If I assume that they have joined and they left, that's what I can guess.
So most of the drug molecules will reach the circulation. 50 mg will reach.
50 mg will reach. Okay. So who said? Vijay Lakshmi, right?
Yes, sir.
Okay. So the point is 50 mg. If you take 50 mg of aspirin, as a oral drug. We discussed about absorption, we discussed about distribution, we discussed about metabolism, we discussed about excretion. Okay, so if you take 50 mg of the drug molecule, what you say is like the entire 50 mg will be absorbed. That's a statement, right? Am I right?
Yes, sir.
Okay, so if you take 50 mg of the drug molecule, definitely not the entire 50 mg will be absorbed. The first point. Okay, I'll tell why. Second point is, if your statement is right, if you are taking 50 mg of the drug molecule as a oral drug, and you found that the entire 50 mg in the circulation. So this is on circulation, and this is site of administration. Site of administration. So if you find this scenario, then we can say that the bioavailability of that drug molecule is 100 percentage. Okay, if the scenario is the second case, you are taking 50 mg of oral drug molecule, the entire 50 mg is present in the circulation, then the bioavailability of the drug molecule is 100 percentage. But very unfortunately, we cannot find any drug molecule with the bioavailability of 100 percentage. You cannot find not even a single drug molecule with the bioavailability of 100 percentage. Okay, so that's why we have to study about what are the various factors which are going to which are going to determine, or which are going to decide the rate of absorption, distribution, metabolism, excretion. Okay, the answer for this question lies on what are the various factors which are going to affect absorption process. And why do we say that no drug molecule will have the bioavailability of 100 percentage? So as of now, the message is that you cannot find any drug molecule with the bioavailability of 100 percentage. Okay, so let me first define what is bioavailability. Bioavailability of the drug molecule is the amount of drug molecule which is present in the circulation. Okay, very generalized, right? So can you tell what is the difference between molecule which is administered through oral absorption, oral drug molecule, and intravenous route? We have different route of administration. One is oral, very commonly used. Another one is also very commonly used method is intravenous injection. If I take 50 mg of aspirin, if I go for intravenous injection, what will be the bioavailability?
So the bioavailability of intravenous will be higher than oral consumption.
I don't need higher, lower. Okay, my question is simple. I'm giving the values, right? So if I use a 50 mg of the aspirin, this time if I go for intravenous injection, my question is what will be the bioavailability of that aspirin? I change the route. That's all.
100%, sir.
It is 100
100%.
percentage. Okay, I show the definition for bioavailability again. Now can you answer? Yeah, just go through the definition given in this slide. So can we say the bioavailability will be 100 percentage? Yes or no?
No, sir.
No, sir.
How do you say no? See, I am not confusing. I hope I'm not confusing you. So take oral route and intravenous route. Same drug molecule, aspirin. Same concentration, 50 mg. Okay, if we go with oral, how much how much concentration of the drug molecule we can observe in the circulation? If we go for intravenous injection, how much? What will be the bioavailability if we go for oral? What will be the bioavailability if we go with the intravenous injection? That's all. Okay, just look and answer for this question. Okay, I think time is up. So we shall continue in the next class. So what I need is answer for this question. You may note it down. Okay. Any more questions?
No, sir.
Okay, so then we will see in the next class. Okay, yeah. Thank you.
Thank you, sir.